innate immunity.
Built in.
Yeah.
It's like built into us. Right?
Built into us. We're born with it. Okay? The other type, which is on chapter 18, we will look at it later, but that one is more you acquire. It's called adaptive immunity you acquire throughout your lifetime. So the big picture, and we will sort of understand this at the end when we look at also the adaptive immunity, which consists of the humoral and cellular immunity. So basically what we have is all of these emerging infectious diseases, or re-emerging infectious diseases that are caused by any of those organisms that we study in this class, viruses, bacteria, fungi, or any of the helmintic or protozoans, or even the prions. And what they're doing is they're trying to go through these barriers that we have. And some of the first barriers that these pathogens encounter are the skin, the mucous membranes, these things that we looked at in chapter 15. They also have to deal with antimicrobial substances. So our microbiome produces those bacteriocins that they are representing an antagonistic environment for these pathogens that are trying to get in. So once they enter these first line of defense, then we have the second line of defense is activated. Then after this, if it's something that is really overwhelming for the immune system, and for the innate immune system, then the humoral and cellular immunity will be activated. But in general, all of them at some point work together to keep us healthy, right? So in this chapter, we are going to look at those physical and chemical barriers in our bodies, but we're also going to look at other factors such as the ones found in the blood, macrophages, neutrophils, eosinophils, dendritic cells, and then we'll look at these processes, the process of inflammation, the complement system, fever, the antimicrobial substances. So we'll look at this here, which is part of the second line of defense. So we'll look at this first, and then this we'll look at next in the next class. So back and forth, we're going to be interchanging these terms while we talk about the innate and the adaptive immunity. These terms that are related to immunity, susceptibility. What is susceptibility? Is when there is a lack of resistance to disease in the host, and so there is lack of immunity. The defenses don't seem to be enough to fight whatever pathogen is trying to cause infection. And when you are immune to something, basically, or having immunity is being able to avoid or to get rid of disease, and using these body defenses. So in all of these factors involved, both in innate immunity and also adaptive immunity, we have this specialized receptors. Remember, I've been sort of mentioning about receptors on the pathogens, those ligands, where they that the pathogens may have around the body of the microorganism or on the surface of the microorganism. So those are called pathogen-associated molecular patterns, okay, PAMP. And then the host has these receptors also that are compatible with those PAMPs, and they're called toll-like receptors. So these things, as I've been mentioning, they have to be compatible, and they are. And our immune system is very effective in recognizing many hundreds, thousands of different types of PAMPs that are out there. Now, we also have what are called cytokines, which are chemical messengers, and these are extremely important also in triggering the activation of some of these cells that are part of, different parts of the immune response. So let's take a look at the physical factors. So we looked at the skin last time, or in chapter 15, the skin, the mucous membranes, all of these things. So the skin is pretty tough, something that is packed. These cells are packed close together, and they have keratin, right? And keratin is something that is pretty tough, a very tough compound or molecule. And so the skin has these layers, the epidermis, the dermis, the hypodermis, that are protecting, the interior of the what they're lining. And so part of the immune response or innate immune response is the shedding of skin. So shedding of skin is something that maybe some organisms may take advantage of that, like the Propionibacterium acne. They feed on that, but it's something that is also a sort of like a process, the shedding of the skin, a process that inhibit organisms to get very comfortable there, right? Because we're constantly shedding the skin. So these are physical barriers. Here we have, again, the skin and everything that is in the dermis, these other structures, the sweat gland, the sudoriferous glands, the hair shafts, the hair follicles, all of these are also play a role in some of the processes of the innate response. So we're gonna see, because some of them are part of the chemical factor or a chemical defense. So again, shedding, dryness of the skin is something that protects us. And you remember some of the vital factors of some of the organisms that they're able to produce. What do they produce?
Collagenase.
Protease, collagenase, hyaluronidase, all of these compounds that are able to penetrate these layers. They dissolve some of these molecules that are making this very strong barrier. But if you don't have a cut, you're probably pretty safe, right? Because again, all of the nature of this particular organ, if you will, is it's designed to basically avoid the passage of things, or easy passage of things. And so in this skin barriers, then we have different types of arrangement of these cells. So for instance, we have tight junctions. You probably saw this in AMP, yeah? Tight junctions, desmosomes, and gap junctions. So they are designed like this to allow certain types of materials to pass through. So the tight junctions, they have this riveted sort of like when you see something like a fabric that has been sewn and that has this type of how do you call that? So this type of
A seam?
seam, yeah. in between the cells, so allows only certain type of material to pass through. And so it limits some of these pathogens from entering and from passing from one cell to another. Then the desmosomes. This one contains these shoelaces-like junctions where they allow for the passage, communication between one cell and another in terms of allowing certain materials that are smaller pass through the spaces that are in between those shoelaces. And then the gap junctions, these ones are more, the function is more of allowing or enabling chemical signals in between cells, and so allows for the communication between the cells. So these are some of the things that are part of, or they get involved in the immune response, because if there is a signal that needs to be communicated to another cell, then that's the job of these junctions to allow and to permit the communication between those adjacent cells. Perhaps sending a signal that there is a foreign, entity that is present, and so that alerts the cells from that danger. And so, yeah, here is, I may have had this before, did I? Yeah, so when we were talking about fungi perhaps, this is this type of fungi that is called Gardner's, not the fungi, but Gardner's disease is when, as a result of getting these wounds from gardening with the thorns and things like that, you get an infection with this particular type of fungi, and so it could get really bad. So it's usually if you have a cut, when you have, or something that is helping penetrate the microbes underneath your skin, then things like this could happen. I mean, I really don't know how this person got like this, but that looks pretty bad. Because it starts spreading, maybe they got a few scratches and stuff, but that got really bad. Another physical factor is the ciliator escalator. And in the ciliator escalator, you have mucus being produced by these goblet cells, and you have cilia, right? And the cilia, between the cilia and the mucus, all of these microbes that are trying to get into our lungs, they get trapped. And then you cough it out, right? And you either swallow it or spit it out, hopefully not on Thompson while you walk into class in the morning. But you spit it out or you swallow it, and the gastric juices just take care of those microbes. But this is one way that our innate immunity, everybody has this, right? Everybody's born with this. And then maybe at the end of your life, or maybe later on, 20, 30 years later after you started smoking, some of these sort of are wiped out, disappear, and you tend to have all of these upper respiratory infections because you don't have this anymore to trap all of these microbes that are trying to get inside of you. Another physical factor, the lacrimal apparatus, right? So the lacrimal apparatus consists of this duct here and this little sac where the tears are produced, and that together with the hairs of the eyelashes, and even the hairs on the eyebrows. All of these protect us from things, including the microbes. You get something in your eye, and one of the first things that happen is that you start tearing, right? Because your eyes are trying to wash off whatever got in there. And so with that, we have the chemical barriers. So the chemical barriers are anything that are bodily fluids, such as saliva, tears, sweat, urine, all of this, and even some of the vaginal secretions. These are chemical barriers because they contain certain substances that are not very friendly to the microbes. And they help wash out things that are trying to or have been able to get into these areas. And so in the skin, we have a chemical barrier. So we have sebum that is produced here, and that sebum, and also in the sudoríferous glands, then you have the sweat. So between those two things, you have an environment that is not very friendly, again, for certain organisms that do not like salt or acid. Our skin is pretty salty and sort of with a low pH. And so organisms that thrive in that type of environment, we looked at some of those in the lab. Do you remember what those were?
The halo tolerant ones?
Epidermidis.
Oh.
Yeah. Staphylococcus. Staphylococcus. Remember how it grew in one particular type of clay? MSA, right? Because of the salt content.
Yeah.
And so it's or one type of genus, because Staphylococcus aureus, epidermidis, and other species, they grow there. That not all of them are able to ferment all types of sugars, but they do. They are able to survive in that type of salty environment. Yeah.
So, tears was there as like it physically washes away the microorganism, but the tears are also salty. So are all the ones that like, cause like the tears, the sweat, and the urine, those so they're physical and chemical, or are they like, or should they be categorized just in one?
So the physical part will be
like the washing,
the lacrimal apparatus.
Okay.
But the tears themselves, those are the chemical.
So like the fact that the tear washes something away, that's physical, but that's
that's chemical.
It is chemical.
That is washing away whatever got into your eye.
Okay.
Okay.
Don't think too much about it.
No.
Okay.
Just the way it is.
Okay, thank you.
These are physical. The skin.
Yeah.
The ciliator apparatus.
Okay.
Other physical things, we have segmentation, peristalsis.
Okay.
Right? Those are physical things because they're moving. And then another thing is what is in those structures.
Okay.
That will be more chemical. So if you have defecation, urination, those are chemical.
The
Okay,
stomach
thank
acid
you.
would be chemical, but
Yes.
the peristalsis and the movement would be physical.
So here are other types of chemical and physical factors. Again, peristalsis, defecation. The rest are chemical. So we have, vomiting is a natural reflux that gets activated when we have something that is not sitting well in our stomach, right? So what do? When you have food poisoning, one of the things that you do is you vomit, right? It's a natural impulse to get rid of whatever's making us sick. Diarrhea Yes, the microbe is changing the absorption rate of electrolytes and water and stuff, but also it causes for us to wash away, to get rid of these things. The vaginal secretions. So you have some of these bacteriocins that are produced by the microbes in the vagina, so making it very acidic or more acidic than other parts of the body. And some of the other types of secretions there, the urine, moving the organisms out, saliva, and they don't only have salt or maybe a different pH, they also have IgA, which is an antibody that is one of the first, which is part of the adaptive immunity, but IgA is found also in saliva and in tears, right? And we'll talk about IgA later, but it plays a role in fighting those, one of the first responders to fighting microorganisms. Like for instance, if you eat something, IgA is in your saliva, so it's one of the first factors that is found by these microorganisms. Again, sebum, earwax. What is the role of earwax?
Prevent colonization by hostile microbes.
Yeah, prevent colonization by hostile microbes, but also, so how does it do it? It's sticky, isn't it? Right, it's sticky, so the things get stuck in there, right? And then you clean it, hopefully, not putting your, those cotton tip applicators all the way because you can break your eardrum. So that's another thing. Again, the low pH of the skin, lysozyme, and it's also present in perspiration, and also in tears, saliva, and the urine. You see how all of these bodily secretions, they contain these things that are basically forming or representing an antagonistic environment for the microbes. The gastric juices, that's antagonistic. Again, if we swallow whatever we cough out, those microbes are not going to survive in the stomach. They're going to get, killed by the pH of those gastric juices.
Except Giardia.
Huh?
So except Giardia.
Well, yeah. Like certain things that they have,
Yeah.
these virulent factors that they're able to just, go in, penetrate, and still cause harm. So for that, we need something that is going to kill them, paralyze them, do something, or prevent the ATP mechanism from, working. So the microbiome, that's part of your immune system. And you remember that little short video we showed at the beginning of the semester where the microbiome is acquired during birth and it changes throughout the life of an individual, right? It decreases with age, but throughout life, it changes depending on the diet, on your interactions with others, or with animals, or with plants. All of these things play a factor on that. And so we have different associations, types of relationships with our microbiome. We have some that live in us and that they're not really causing any harm to us. They're actually providing benefits. And the way that they are in our body, or at least the ones that protect us, they are causing that antagonistic effect, or they are occupying a niche, a space, so that other organisms do not come and take over. And so, so that's one of the main things. But again, we have a lot of commensal microbes all over our bodies, in our mouth, in our skin, in our hair, everywhere except in our blood and our brain, right? So once the microbes are able to penetrate these first lines of defense, which are the physical and the chemical barriers, then they are, there's something that triggers for the second line of defense to be activated. And so we have the complement system. You guys are familiar with the complement system? Did I mention the complement system at some point? Okay, the complement system are a series of 9 proteins that are circulating in your blood, okay? And these proteins get activated usually when there's something invading or that has taken over in your body. And so they could be activated in 3 different ways. There's 3 different pathways. The outcome of those 3 different pathways are the same at the end. It's like going to Queens Plaza and then maybe I'll go through Queens Boulevard, but then someone decides to go through, I guess, Thompson and go around, so that will be another way, and someone else decides to come through Manhattan. So 3 different ways, but they all get to Queens Plaza, right? So the complement system, they are labeled here, as you see here, C for complement. Complement system 1, 2, 4, 3, 5, 6, 7, 8, 9, okay? So we're going to look at the 3 different pathways, the classical, lectin, and the alternative pathway. You don't have to remember every step of these pathways. This is the classical pathway. So here's what happens. You have a microbe, right, that somehow it was able to get into the body of the host. And so remember those ligands, those molecules on the surface? Okay, so those are antigens, something that is specific attached to the pathogen. And so when that microbe is in the body, we have these antibodies that are produced by B cells, which we're going to see in the next chapter. They are circulating throughout the blood, right? Looking for things to see if they recognize something that is not supposed to be inside. And so they recognize the antigens and they attach to those, they recognize the microbe by looking at those antigens and attaching to them. So an antibody is Liz and monomer is in the form of a Y, so it has 2 binding sites. And so they attach to that microbe. That triggers for the classical pathway of the complement system to get activated. So what happens is that you get a complement system 1 protein comes and it triggers the activation of complement system 2 and 4. So they split, and then you get 2A and 4B. So this splits into A and B. Same thing with this. So part of A joins B, 4B here, and this combination here triggers the activation of complement system 3. Complement system 3 is key in all of these pathways, because once this happens, they're gonna come and attach here. I did mention at some point the boot, right? Remember the boot when you haven't paid your tickets?
Mm-hmm.
Yeah, okay, here's the boot, okay? So, the complement system will not be activated unless that boot is here, right? Now, several things can happen, or three different outcomes can happen as a result of the complement activation happening. You have opsonization, which is basically the tagging, the labeling, the marking of that pathogen for destruction. The other one is cytolysis, and the next one is inflammation. So let's look at the other two types of complement activation. Again, you don't have to remember all of this. Just know that in this one, the 4 proteins that are involved are complement system 1 through 4. And that complement system 3 is the one that triggers for these 3 things to happen, any of these 3 things to happen. Here's the alternative pathway. This one involves complement system 3 attaching to these other factors, BDP. These BDP factors or molecules are circulating also in the blood. We have a lot of things in our plasma. So these 3 are circulating in our blood, and they are specific to certain types of antigens on the surface of a microbe. So this one will attach to these 3 different sites on the microbe, and that triggers the activation of the complement 3 molecule. And so the complement 3 molecule, once activated, it triggers for any of these 3, like what we saw in the previous one, right? That's the last type of complement activation. This is called the lectin pathway, okay? Because it involves carbohydrate-containing molecules that are complementary. They are these molecules that are complementary to carbohydrate molecules that are on the surface of the pathogen. In this case, lectin is attaching to what we see here as mannose, a carbohydrate. So these lectins are also circulating in the blood, and they attach to the specific sugar that is making the antigen on the surface of that pathogen. And so that binding here will trigger the activation again of complement system 2. Here we bypass complement system 1. So 2, 3, and 4 are the ones associated with this complement system. But again, these are the outcomes. So let's take a look at those. So this is another type of process where this basophil or mast cell is activated by the complement system 5, which helps in the degranulation of this mast cell and causing for the mast cell to release all of these histamine mediators. So this is something that helps or that is activated during the process of inflammation. Again, this is like a side kind of process that involves the activation of this mast cell by complement system 3 and 5. So here are the 3 outcomes. You have opsonization, which is the tagging, right? So that involves immune adherence. And what that does is that by opsonizing, you are marking, labeling that microbe for destruction. So now the phagocytes, the wandering phagocytes that are going around in the body, in the blood, searching for intruders, they will come and they know that that's the one that has to be that cell that has been opsonized has to be destroyed. So that's the one that contains the boot, right? The other thing that complement system does is membrane attack complex, causing cytolysis. So these are bacterial cells. And what the complement system does is that it causes, let me see if I have it here, yeah. So it, basically you have complement system 6, 7, 8, and 9. They come together close to one of the transmembrane channels on the membrane of the pathogen, and they force it to open, and that cell is going to leak out its contents. So this is the process of cytolysis. It's going to lyse that bacterial cell. Okay, so that's another thing. And the third one is inflammation, which we're going to see in a few minutes. Inflammation is activated, and several factors of the blood and the immune system are activated here to rush to the area of infection and trying to resolve whatever is going on in that area where the pathogens were able to enter. Okay, you guys okay? You need to get up and I see some of you like, okay.
So I have just keep in mind that if you know the lectin pathway, or if you know the classical pathway, you pretty much know the lectin pathway.
Okay, so the classical pathway involves 1, 2, 3, and 4, right? And when 2 and 4, you don't have to remember that, but just involve those 4 molecules or 4 proteins. And in the end, the goal is to activate complement system 3. Once the complement system 3 is activated, which is activated after 2 and 4 split, then you get the any of these processes happening. Then in the alternative pathway, you have the BDP proteins that attach to the antigens, and that triggers for the complement 3 activation. And then again, the outcome is the same. And then you have the lectin pathway, which are these lectin molecules that are circulating in your blood and that are specific to carbohydrate mannose, in this case mannose, but it could be other types of carbohydrates. So they're very specific. And that also triggers for the activation of, in the end, complement system 3. And triggering either opsonization, cytolysis through the membrane attack complex, or inflammation, okay? Now here are the cytokines. So we have 3 main different types of cytokines. And these are, again, involved in communicating. Hey, there is an intruder here, there's a virus, and the response could be different. If it's a virus or if it's a bacteria, there are different chemical mediators that are activated in order for the cells of the immune system to get activated. So, the first chemical messenger is the autocrine, where a cell of the immune system secretes one of those cytokines and auto-stimulate itself. Okay, so this one is producing the cytokines. Here is releasing the cytokines, and it has receptors on the surface of its own cell, and those cytokines are going to bind with the receptors outside. And you would think, but what's the point? I mean, why? Well, this is a way on how they display that there is something going on. Maybe there was something that invaded this cell, so they were able to release these cytokines, and now by attaching it to these receptors here, they are displaying it, and other types of factors in the immune system will recognize that, okay, as a, like when someone is caught captive and they're not able to get out, and maybe they put like a little white flag or I don't know, something to let people know that, hey, I'm captive here, please send the police or something. Okay, the paracrine. This is where one cell secretes the cytokines, but they don't autostimulate those cytokines. They travel to an adjacent cell, a cell that is close by, and then that cell will respond in some way by producing maybe another cytokine that something else is going to recognize. And then the endocrine type of messenger is the one that is secreted by one cell, and then the cytokines, they travel far away from where they were and affecting. It's received, the message is received by a distant cell. So remember the pyrogenic effect of gram-negative bacteria, that the bacteria kill, and then those lipid A and O antigens, these things, these factors on the membrane of the gram-negative, they're not, well, all of the cell is destroyed, but the fact that these things are still present in the cell that destroyed it makes that cell, that host cell, release cytokines, interleukin-1 and 2 necrosis factor. So that cell is releasing that, it's gonna go to the circulatory system, and the message is gonna be carried all the way to the brain, triggering the thermostat to change and for the person to experience fever. Even after they've been taking antibiotics for a while, they experience fever because those antigens, those particles of the gram-negative bacteria have triggered for the sort of reshuffling of your body temperature. Here's a cytokine that is only released during viral infections. Interferon, alpha, beta, and gamma are released only, so that name, interferon, is a type of cytokine that is only produced by cells who have been infected with a virus, okay? And so when the cells are infected with the virus, here is a phagolysosome, okay? The virus is destroyed, so now this cell is producing interferon and is releasing it. So there are three things that could happen here. You have those interferons signaling another cell, okay, to destroy the RNA and reduce the protein synthesis process. So that way the viruses, remember the viruses only use their host machinery. So that way this cell will not, or will sort of make it difficult for the virus to replicate its parts in it. Another type is the interferon reaching a cell and telling them, the master says, you know what, you need to kill yourself. Because, yeah, you need to kill yourself because otherwise you're gonna get a virus soon, because there's a virus going around, and it's gonna come and use you to multiply and stuff, so just go through apoptosis. And another one is the activation of other cells, of other immune cells. So these things are very active. These viruses are very active. when you get a viral infection, they're very active when you're active. When you rest, these processes of the virus being able to do this, or infecting other cells, sort of slows down because you have your immune system, all of these interferons, sending all of these messages, and your immune cells sort of working hard to avoid that virus from replicating. Okay. You familiar with this?
Yes.
Okay. You too? And
Very.
okay. All right. So we're not gonna spend a lot of time on this, but these are part of your immune system as well. So you have that hematopoietic stem cell, which is a naive or a virgin type of cell that could differentiate into different types of cells. So you have a myeloid stem cell and a lymphoid stem cell. So the myeloid stem cell is going to differentiate into different things. You have a megakaryocyte that will differentiate into platelets. Okay, very important. In what type of process? Clotting. Right. And so you also have erythrocytes. So all of these are derived from the myeloid stem cell. The erythrocytes that carry the oxygen to all of your tissues, the mast cells, which are involved during a lot of the allergic reactions, and the myeloblasts, which will differentiate into the ones that you probably saw a lot in AMP, right? And so basophils, neutrophils, eosinophils, and monocytes, very specific to the types of pathogens that are infected and their response. Now the monocyte, which is the one with the largest nucleus that takes over most of the cell, is going to differentiate further into macrophages, very important because these are some of the first, these are the Shoshanas of the Shoshanas of the immune system. The MTAs and the paramedics, they are some of the first ones.
That's funny.
Then dendritic cells. They're not dendrites, really. They're called dendritic cells because of their shape. They have these extensions. Now, the lymphoid stem cells, these are the ones that are associated with cell-mediated immunity. So we have the natural killer cells, which is a big lymphocyte, and we have a small lymphocyte that differentiates further into B cells and T cells, okay? And so we are going to look at those again in the next chapter. However, just a brief reminder of the blood. So you have about 55% of plasma, and then you have a larger percent of red blood cells in comparison to white blood cells, which are only this much in comparison to how much we have of those erythrocytes. And a couple of types of conditions that are exhibited when some of these cell counts are either very high or very low. So an increase in leukocytes, meaning those white blood cells, that means that you have a bacterial infection. And that's usually, the doctor has ways of looking at this by, touching certain parts of where the ganglia, these lymph nodes. And leukopenia, decrease in leukocytes, and that's for someone who is like maybe HIV and has reached maybe the stage of AIDS, very low, white blood cell count. So again, the neutrophil, eosinophil, and basophils, you distinguish them mainly because of the nucleus, the log nucleus. The neutrophils, quite important also in helping Shoshana and those responses. And so these are some of the things for blood. mast cells, they are similar to basophils, okay? But mast cells can also migrate within tissues, like to areas where you have some kind of inflammation, something where they're needed. We'll look at those a little bit more in the next chapter. And the natural killer cells, which again are part of the lymphoid stem cell, okay? They are derived from there. And what they do is that they kill. They basically destroy cells that are not, so they are these MHC complex molecules. Did I mention MHC? Or not yet? Okay. So because that's part of like the next chapter. So let's say that you have, okay, this bacteria is going to be engulfed by this macrophage, right? Because it had that antibody attached to it, right? And so now, and it has been opsonized. Remember what opsonization is? Huh?
Tagging?
Yeah, the tagging. So it's been tagged, so now that macrophage knows that it needs to go and grab that and destroy it. Once inside, we have a lysosome. So once this comes inside, it's inside a lysosome. And that lysosome is gonna fuse with this. You also have these other molecules called the major histocompatibility complex, MHC. There are two types, MHC1 and MHC2. Macrophages, neutrophils, B cells, dendritic cells, they exhibit MHC2, okay? And so what they do is when this pathogen is destroyed, so now it's like, it's destroyed. So now the MHC molecule is going to take some of the residues from here, okay? And it's going to put it here. So this is a receptor that is going to be identified by other cells of the immune system to be destroyed, okay? So that's the major histocompatibility complex, which is a molecule that joins part of the waste of the microbe that is causing the infection and is displayed on the surface of that macrophage. Anyway, so here, the natural killer cells, they recognize an MHC1, which are more involved in MHC1, more involved with T helper cells, such as cytotoxic T cells. So, okay, and what happens here is that you have a healthy cell is displaying, this is the healthy cell, is displaying an MHC1, okay? It is something very similar, something that contains some abnormality that is happening, okay, together with that protein. So it's displaying an MHC1, in this case it's a receptor, and so if it's displaying it, the natural killer cell has also a receptor, it's called an inhibitory receptor. So it's gonna leave that in normal situations, when this cell is normal, doesn't have any pathogens inside, such as a virus or maybe a cancer cell. these cells are gonna be left alone because these are healthy, right? But in this case here, the natural killer cell is looking at an infected cell of the same type that is not displaying the MHC1. The fact that it's not displaying the MHC1 means that it has been infected, okay? And now the natural killer cell gets activated and release these chemicals that are going to destroy the cell, taking it through cytolysis. The chemicals are called perforin, so they just release these chemicals that are gonna make holes in the cell that is infected and it destroys it. So again, the MHC1 will, and actually MHC2, we'll see them in chapter 18 a little bit more. So here are the monocytes with the large nucleus in a blood slide with the red blood cells. And they will become macrophages. Remember, they could become macrophages or dendritic cells. And so here are the macrophages, and they just go and eat whatever doesn't belong in your body. We have wandering macrophages, but we also have some that are more sedentary, but their role is the same, to get rid of whatever it doesn't belong in your body, in terms of pathogens. Okay, so here's another process that is done by neutrophils, and this is called oxidative burst. So when we have a pathogen inside our body, we have neutrophils getting activated and basically destroying. They go through this process of the pentose phosphate pathway where NADPH is produced, and we get the production of superoxide molecule. Superoxide dismutase, remember that I said that aerobic, facultative anaerobes, and aerotolerant anaerobes are the three types of organisms that need to produce superoxide dismutase and catalase when we did that experiment in lab. And I said, and we also produce it. This is why when we add some of that hydrogen peroxide in a cut or something, we get bubbly, right? Because we are able to produce that too. So this is what is happening, and what these neutrophils do is that they just basically destroy those pathogens using hydrogen peroxide, okay, or through oxidative process. All right, questions? I know that I'm going 100 miles per hour. Questions? You good? Okay, yeah.
I guess for the exam, will you be asking more questions about kind of the processes and functions, or like for example with the flow chart that demonstrates like how all the white blood cells evolve? Like, do we need to memorize like everything on that?
No.
It'll be more function oriented, similar to the
More
past
functioning,
exams.
how they're involved in
Yeah,
other
how things work
processes, and
together.
you're gonna see, when we look at inflammation, we're going to see some of the neutrophils and the macrophages involved in that process, so that type of thing.
Right.
So
okay.
more on the process, not so much on,
Yeah.
you know.
So
Yeah,
no,
this is something that you already knew, right?
Right,
From
yeah.
A&T. So we're kind of
but
bringing
the
it
questions
up.
will be asked in context, basically. It won't just be like.
Exactly, yeah.
Okay, got it.
Okay, so this is also part of the second line of defense, fever. So what I was mentioning earlier, the gram-negative bacteria and the pyrogenic response. It causes for the release of pyrogens, these molecules that contain some of the remains of the gram-negative bacteria that has been destroyed. And so they are, I don't have it. I think I have it So basically, what we have is you get infected with a gram-negative bacteria. You start taking antibiotics because you're feeling all of these things, fever and whatnot. And then 5 days later, you're still, having fever and stuff. Because when the bacteria is destroyed by our cells, let's say the phagocytes, and they're destroyed, and some of the remains of the gram-negative bacteria that are on the cell wall, the LPS, the O antigen, the porins, all of these things, they trigger the release of histamines. And the histamines in this case are interleukin-1 and 2 monocrosis factor. Okay? Interleukin-1, 2 monocrosis factor. So they get released and they go into the bloodstream. In the blood, they're able to travel all the way to your brain, to the hypothalamus, where they cause for you to shiver and to experience fever. And there's a change in the body temperature. So that's what is called the pyrogenic effect. This is basically what is happening there. Hypothalamus releases prostaglandins that reset the hypothalamus to a high temperature. The body increases the rate of metabolism and the shivering, and so that raises the temperature of your body. And you also have vasodilation and sweating. So if you don't take care of this either with medication or cold compress or whatever, you can end up even dying if the fever is too crazy. You can go into crisis. Okay, so, but this, what the immune system is trying to do here, or fever is trying to do, is burn those suckers. They're trying to burn, they're trying to respond to the presence of those antigens in the gram-negative bacteria that is causing the infection. All right, inflammation. Did you guys do inflammation at all during, yeah? Okay,
Yeah.
so you. So that looks familiar, right? So you get a cut, right? What?
It's like you keep like the whole semester you keep talking about ADP, right, or whatever that is, and we didn't do it because we're vet tech,
Yeah.
but we have no idea. Anytime you say, "Oh, you said,"
Oh, you're vet tech?
yeah.
I didn't know you were vet tech.
Yeah.
You're not vet tech.
No,
I think
there's
it's just only
Us
us
three,
three.
maybe.
Yeah.
And
There's
Isabel.
So that's- the whole semester is just funny, like, "Oh, you see an AP or whatever."
Our AMP class, he
AMP
had his own
is cool.
agenda. I don't know. We didn't do all the. Like, I think he skipped some of this.
A lot,
Okay.
a lot of the stuff from this PowerPoint that
Yeah.
I know is from a clinical vet tech course.
Ah,
So like
okay.
you will learn it.
Not yet.
But yeah, our AMP was not the human one.
Maybe I'll suggest it to the coordinator of your AMP. Because I can see, like, I mean, if I don't ask, I wouldn't know. I thought this was covered because those classes are required for this class, so that you're supposed to have this already.
I
So
don't.
when I talk
I
about
covered
it,
the basics of it,
vet-
maybe, but.
The vet tech, the vertebrae anatomy, because that's what it is. It's not anatomy and physiology of people.
it's just
vertebrae
anatomy.
Yeah.
And
It's
it
bones,
it
muscles.
should not it should not be a precursor to this course. Like it just we don't have a foundation. Like I talked to you about it
Yeah, yeah.
at the beginning of the semester, and like I'm getting through it. We're all getting through it, but
Oh, you're
I-
vet tech too?
I'm
Yeah.
I'm in the program.
Okay.
But I feel like there should be a conversation of the
Well,
departments.
I will bring it up. I will bring it up to them.
Thank
Because
you.
it's not fair to you guys. I mean, the students are struggling, and I mean, you kind of take these courses. We're assuming that you cover the curriculum,
Yeah.
you know, this stuff and nothing. Anyway, so I'll talk to someone. I don't know how much, but hopefully I'll make waves.
Thank you.
Okay, so here at the skin, you get a cut with a knife, you're cooking, and that knife has bacteria on it, right? So the bacteria is able to penetrate, right? It's cutting some of the small blood vessels, and that's why you get a little blood coming out, right? Hopefully you will get, you'll have no problems with clotting, and the bleeding will stop and whatnot. But and inflammation is an immune response that is so, I think it's so cool the way things happen. Because the whole process is trying to avoid the spread of that pathogen into the rest of the body. One of the first things that happen is that there is, like when there's a crime scene, that they put those tapes around. So here's the crime scene, okay? And so everything is isolated there, okay? Where the bacteria have penetrated. And you have the cells that have been ruptured or damaged. They release these chemicals, prostaglandins, histamines, kinins, leukotrienes, cytokines. They release these things, and the role of all of these chemical mediators is to alert other parts of the immune system and saying, hey, we have these microbes here, we gotta take care of this. So, first thing, isolation of that area. Notice, and inflammation could be it could be anything. In this case, I'm talking about a knife that you gotta cut, but it could be a mosquito bite. That causes inflammation too. Tell me what you notice when you get a mosquito bite in the summer.
Swelling, itching,
It's itching.
swelling.
What else?
Sometimes
Swelling,
swelling.
redness.
It's redness. It's bumpy, right? So you have edema, you have erythema. So you have all of these things. And why is this happening? Because your blood vessels are enlarging, right? To allow the cells of the immune system to rush to that area, okay? And to take care of whatever is going on there. The phagocytes, the neutrophils, they rush to that area, and it's easier for those guys to get to that area if the blood vessels are sort of enlarging in diameter, because it's easier to get there. Okay, so you have blood clot forming at the ends of the little blood vessels, so that you don't get, unless you have something like Streptococcus pyogenes producing this streptokinases and, not allowing you to clot your blood, or Staphylococcus aureus or something like that. Okay, so you get that abscess there. There's vasodilation, again, the enlargement in diameter of those blood vessels, and here's what's happening. You have, this is the blood vessel, okay? When the diameter of the blood vessel enlarges or becomes bigger, what happens is that you get more cells being transported to the area. Let's say that this is the area where the infection is happening. You see the bacterial cells. So you have monocytes that are sort of attached to the walls of the blood vessels, okay? You have neutrophils that are rushing to the site of the infection, but they need to get to that area, sort of cross that blood vessel. And what happens with the blood vessel is that not only they're getting wider in diameter, but they're also getting liquider. So they will squeeze through those spores to be able to get to that area so that they can take care of those bacteria that are causing the infection. Macrophages are doing their thing, going and grabbing, those bacteria, getting rid of them. And so that process of the neutrophils going through those pores is called diaphoresis. The process of the monocytes attaching to the walls of the blood vessel is called margination. Phagocyte, monocytes. What else is happening here? Okay, when these guys are done with their job, say that they clean up the mess, they got rid of all of these bacteria. What happens here is that they start dying. They're not needed anymore. They start dying and they produce pus. Okay, that pus is full of like waste and a lot of things that are not needed. And that sort of goes back to the interstitial fluid. It's gonna go back to your lymphatic fluid or combine in there, and it's going to be cleaned up. But this pus is basically your macrophages that have died, okay? And so this is what happens. Once this abscess has been reduced because now the waste has been recovered, has gone to wherever it needs to go, and the process of healing will start by producing, again, a blood clot and then a scan. Notice that when you get a cut at home when you're cooking, maybe it takes like maybe 4 or 5 days for you to heal. But imagine this process, it's so perfect. I mean, the way things happen, I'm always amazed by, our body, the way that we respond to these things, and that we're able to heal. Usually we're able to heal and repair the tissue that has been damaged. So that's the process of inflammation. And remember, inflammation is triggered by the activation of the complement system. Remember that? So these things that we cover are triggered by the complement system. So some of the things that happen during inflammation is also sometimes you may get like a display of a rash, right? Varicella, chicken pox. This is one of the characteristics of those types of diseases. But in general, the release of all of these chemical mediators cause the redness, the edema, the pain, the heat. So again, even with a mosquito bite, you touch it and it's like warm. Why? Because you have all the rush of the blood and all these things going into that area. And a lot of them, what they cause is the release of histamines, vasodilation, permeability of the blood vessels so that these blood factors or cells can go through those pores. Here's your lymphatic system, which I believe the EMP people did. You probably didn't do the lymphatic system? No? Okay. So is where, so you have, it's like a super highway, similar to the circulatory system, it's all over your body. And then you have sort of like these super cities, where you have accumulation of these immune cells that will get activated and will be produced during an immune response. So the T cells are produced and mature in the thymus gland, which is right on top of your heart. That looks like a chicken breast, open like that. And the size of that organ starts reducing with age. By the age of 80, it's like almost gone completely. And part of the reason why you have, your immune system is not as strong, because it's sort of depleted with time. But it works in conjunction with the circulatory system, because the circulatory system takes all of these things, all of the these factors into areas of infection. Now, once the work is done, and of course, in the lymphatic system, you also have these immune cells that are going to be produced and run into these areas of infection. And when you are sick, like if it's a bacterial infection, this is when the doctor will sense some of these nodes being sort of enlarged.
Can you see the lymphatic vessels? Are those like physical things that you can see in a person?
Yeah, they're not as obvious as the circulatory system, but yeah, it's a network of vessels. So it also takes, again, whatever waste is produced during all of those processes of destroying pathogens, it takes that into the process, or the structure is more like a vein, okay? And so it flows from the interstitial fluid and the cells. That stuff flows back, and again, it's taken care of, like almost like one of those environmental companies that get rid of our chemical waste and microbiology in the proper way. But it's very well designed. Imagine our body doing all of these things as we talk. And it's, I don't know, I'm always very amazed by that. So here are both the circulatory system and also the, bringing stuff into the tissues. And this one is taking that fluid or that waste away from the tissues where it has been, where it collected whatever was the waste from process of destruction during inflammation. Okay, so second line of defense includes fever, right? The complement system, and we have phagocytosis. And these are phagocytosis, right? These are phagocytes, which are monocytes that turn into these guys. And so we have fixed macrophages, like the one we saw on that blood vessel, fixed macrophages, okay? And wandering macrophages, this one here. Okay, they have these extensions, pseudopods, that allows them to get or helps them grab those pathogens and bring them inside. Now they destroy, that's their job, to destroy whatever they trap, eat them, get rid of them. This is a phagocyte, and what we're looking at here are 3 different types of receptors. And this is just to illustrate that phagocytes can have many different types of receptors that have specificity to the type of pathogen antigen, or antigen on the surface of the pathogen that they are identifying to destroy. So these receptors here on the surface of this macrophage, they have different binding sites. So for instance, this lipopeptide has this shape. This will not fit here, will not fit here. So again, it's to show specificity. And we have thousands of types of recognition sort of patterns that our cells can do for the different types of diseases that we're exposed to. So this again these are part of the pathogen. It could be, this is flagellin, so it's part of the flagella of, let's say, a bacteria or a protozoan. And so those are the pathogen-associated molecular patterns, or PAMs. And these are the toll-like receptors, okay, or pattern recognition receptors, and they have to bind. They're very complementary to each other. So here's what I drew here, not quite as nice as this, but here's the pathogen, okay? It has been, here is not showing the antibodies, but basically the pathogen here has been identified by this macrophage for destruction. So once it goes in, we have the lysosome providing digestive enzymes to destroy that pathogen. And once it's destroyed, it gets rid of that, but also when you have, or usually what the macrophage also does is that part of this material joins the MHC2 and is displayed on the surface of this macrophage. That attracts a T helper cell. Is being recognized by a T helper cell only because it's displaying part of that pathogen that was destroyed. There are some microbes that could evade phagocytosis. Some of them are able to inhibit phagocytosis by producing a capsule. That was the first thing we learned about what capsules do. So Streptococcus, no, not that one. But let me see, these are all. Klebsiella pneumonia has a capsule. Klebsiella aerogenes also, or Enterobacter aerogenes also have a capsule, and that inhibits the phagocyte from engulfing it. But these other guys, Streptococcus pyogenes, Streptococcus pneumonia, they inhibit the adherence of the pathogen with the receptor on the surface of the phagocyte. So there is no adherence, so there's no vacuole being formed to engulf it. Staphylococcus aureus produce leukocidins, which are chemicals released to kill the phagocytes. Other types of microorganisms are able to survive inside the phagocyte. So they can escape the phagocyte. Or this one here, Listeria, can also lyse the phagocyte. So by destroying the phagocyte through a membrane attack complex, it will, so just making pores on the membrane of the phagocyte, they will destroy it, and that way the Listeria cell can continue, taking over. Some will be able to survive the phagolysosome. They can escape it. They can escape, and it's not destroyed by the hydrolytic enzymes inside that the phagolysosome. Others will just survive, so those are other examples. This is Leishmania, which is a trypanosome that is similar to the trypanosome. And it can cause cutaneous leishmaniasis or visceral leishmaniasis in tropical areas. It is transmitted by a sandfly, and so cutaneous leishmaniasis causes these boils in the skin. When you open one of these are caused by the action of phagocytes and different types of immune cells trying to destroy these Leishmania parasites. If you open one of these, you'll find the parasites in there. But this is something that will be caused by these guys. Here we have a white blood cell, a monocyte, with immature leishmaniasis adults or parasites called mastigotes. Here's when, that process of inflammation, if everything goes well, goes the way it is on those 2 slides or 3 slides. But sometimes you have some pathogens that may be able to sort of stop that process, the successful process of inflammation. And that's cause here, an example of that is a granuloma. Can you talk about TB? Granuloma? You spoke about TB.
I know, I think.
Oh, I'm sorry, it wasn't you, it was you. Sorry. You're like, well, okay.
No, okay, so yeah, you have what was interesting is that another thing about it is that you do have some macrophages who send out signals as they have the TB inside of them. And then other macrophages and lymphocytes, they wall off the area. So you have the initial site of active TB somewhere in a lobe, and then for years you'll have, what I was interferon gamma. They're being secreted so that way it suppresses the activity of the TB to multiply, so it just stays dormant in this granuloma. And then if it becomes active, then it multiplies, it goes open, and the granuloma becomes a liquefied cavity.
Yeah. So you can have the bacteria in this wall because they are protected from the immune system, and so they could become active at some point in the future for people that have tuberculosis. So this is what is called an unsuccessful, unresolved inflammation, okay? Because this is not completely, destroyed. As we saw in the inflammation, where you have this process of healing, but here is not. And here's another type of sort of inflammation, type of chronic condition where filarial nematodes trigger for the accumulation of that fluid, waste in the lymphatic system, and people accumulate some of the lymphatic fluid because the worms are actually sort of clogging those lymphatic ducts, and you get this sort of inflammation that is not resolved. Okay, last slide. Physical defenses: skin, mucous membranes, endothelia, shedding skin, peristalsis, segmentation. Okay, chemical defenses: sebum, saliva, mucus, gastrointestinal fluids, urine, tears, serum, and vaginal secretions. So any bodily fluid, those are chemical defenses. The microbiome, our microbiome, it provides antimicrobial peptides, so things like dermicin, cathelicidin, defensin, cystatins, bacteriocids. All of these are enzymes released by our bacteria, the bacteria that live in our body, and that help us sort of maintain opportunistic pathogens away. Cellular defenses include red blood cells, white blood cells, platelets, all of these cells that we looked at associated with blood, granulocytes, neutrophils, eosinophils, basophils, mast cells, natural killer cells, monocytes, macrophages, dendritic cells, the complement system, and cytokines. Then we have part of the part of the second line of defense includes phagocytosis with the pathogen-associated molecular patterns, toll-like receptors that they got to complement each other, the phagolysosome that destroys that, and that we'll talk more about the MHC later in the next chapter. Fever, second line of defense, changing the body temperature and triggered mainly by bacterial infections and inflammation. Having all of these mediator chemicals triggering the activation of the different cells associated with the response to the area where the inflammation is happening. And these are signs of inflammation. Questions? This chapter is not so bad, is it?
Professor, I have a question. So, complement system, right? Complement all the cellular defense to classify this immunity. It looks like a kind of like a acquired immunity because the cells are modified.
Acquire? Not yet. Acquire immunity would be, so everybody's born with this. Everybody's born. Innate immunity, non-specific defenses of the host. Everything here is innate immunity. You have first defense, second line of defense, all of these are part of it. The cell mediated, we'll look at it next time. That's chapter 18. Okay, so give you a peek until I'll make it available. Chapter 18. Okay, so here's the entire. We're gonna go and just. So here's the whole picture of the adaptive immunity. You have the humoral and the cellular immunity. You have the B cells, and we'll explain what happens here, and we have the T cells. And how sometimes these T cells have to come and help out the B cells. And then we have these other things happening. But we'll talk about antibodies and the type of antibodies. Where are they produced? Okay. What do they do? You know, here's the, see, I was talking about the MHC, Major Histocompatibility Complex. It's, look, it looks like a receptor that has come out from here with an antigen fragment. It comes out here and it displays part of that antigen. So now the T helper cell recognizes it and it's like, oh, okay, this one needs help. And so it comes and it triggers for the B cell to be activated. When the B cell gets activated, it produces antibodies and it also produces memory cells. So this is just part of what we're going to see, okay? I'll make it available so you guys have it. I don't know how much we're going to, I usually like to cover up to chapter 19. So what I can do is, I'll have, we'll see. Friday is the last day, okay?
For the lab exam, how many questions is it gonna be?
About 20 stations.
And then we'll just have to like.
So about 20 stations, you'll have, let's say that you have a, you have 3 load agar plates.
Okay.
One with alpha, gamma, beta.
Okay.
And I can ask you what type of, you know, which one is beta or which one is, what type of organism will cause that. So when we did that, do you remember the type of organism that causes beta hemolysis? Bacillus Staphylococcus aureus. Yeah, so, and maybe I should have remind you more times about taking notes. You see the importance of taking notes? Because we go over this when we look at the results. And if you don't take notes, you're not gonna remember.
Sure.
It's too much information. But if you write it down, you can always go back and say, okay, beta hemolysis, let me see the organism. Okay? Yeah.
So like, for me, name the organism, because I was saying that Bacillus, like, that causes beta hemolysis, but like, one of the organisms that could have been my unknown? Like, it has to be specifically the one we tested?
The one we tested.
Oh, okay.
The one we tested. And I'm not going to ask for a lot of like specific genus and species. You know, maybe something that is very obvious. Like we did the Manitou, or maybe we did the Macankey, or maybe we did the EMB. What did we do for the EMB? Do you remember?
Was it E.
Which one
coli
and how does it look like?
for EMB? Wasn't it like
For E. coli
Oh,
and EMB?
it got metallic? Right?
Metallic,
Oh,
right?
yeah, yeah.
So if you see that,
The ear.
and
one
if
of
you
the plates.
if I ask,
Yeah,
okay, what type of organism will
Yeah,
exhibit this kind of
eosinophil
reaction?
blue.
That metallic green, we didn't see, we only saw it once, right?
Yeah.
But it's very nice, and so it kind of stays here. And you can either tell me an enteric or E. coli. E. coli will be best, but that kind of a question,
Okay.
alright? If I show you two plates
20 station.
with starch hydrolysis, which one is positive and which one is negative? You should be able to identify that.
Well, then-
What was this starch broken down into?
Lactic, no,
A disaccharide. Which is the disaccharide?
lactose, no. Maltose?
Uh-huh.
Maltose?
Maltose. So these are the type of questions, okay?
Bless us all.
And it'll be like the anatomy practicals where you like go around once and you have like a minute at each station, and then you go around the second time, you have 30 seconds. Okay.
Like for the extra time, do you?
So we're probably gonna have to set up something, maybe after. I have it set up in another room or,
Like sometimes, in my exams I've been taking lately, sometimes they'll do like, okay, at the end you can go back around again or something.
will that work for you? Yeah, we could do that,
Okay,
we could do that.
so I think maybe that's easier.
Yeah, it's just that everybody else needs to look at their stuff. If it's,
Like after everyone's done.
yeah, maybe somebody could take like a half an hour break.
Oh, for the unknown.
Yeah, cause they need to look at the unknown, yes. It's like 40 max. Because you go around
Show one of the-
20 questions, 1 minute each, so it's like 22 minutes, and then you go around for, another 10 minutes, for half a minute, yeah, it's not a long time, yeah, we'll figure it out. Alright?
Oh, Professor, so the question you said like bacteria shape, are you gonna like put it in a microscope?
Yes. So we will have at least 3 microscopes, I think. And I wrote down on what I posted, you should know morphologies,
The
Okay.
arrangement,
arrangements,
types of
Okay.
staining.
and you read the questions before you answer.
Will it be 2 questions per station?
Sometimes.
Okay.
Sometimes it will be 2 questions. So I'm not asking, identify this, or is this a positive or a negative? Sometimes I may be asking something else. So read the question, look at the specimen, then answer the question. If you have a question that maybe you don't understand, you're unclear or whatever, maybe somebody moved the microscope. Professor, call me right away. If you wait for the minute to pass by, I will not come by. You need to call me right away, because that kind of is like, and I'll probably be there along with, because the technicians are so busy. So, the microscope may be a type of staining, so you should be able to differentiate between what is a grand reaction, what is a negative stain, right? Based on what you're seeing, okay? Maybe identify the structure. it could be a capsule, it could be an endospore, okay? And those things are in your book, you can look at them. Pretty much
What
done.
about the antibiotics? We are supposed to give them-
I may have a question on antibiotics, just, you know, if you just look at even the PowerPoints, what is the area of inhibition, right? So if you look at the area of inhibition and you have 5 different antibiotics and you see 5 different diameters, which one is more effective? Was it the one with the shortest diameter or the one with the biggest diameter? Okay? If it doesn't have any diameter at all around the disc, what is that? Resistant. Okay? So those kind of questions.
Not the name of the antibiotic.
No,
It was so tiny.
no. I mean, if anything, we'll probably have it there, but, no. I mean, we're not gonna ask you something that is very possible for you to see. No, the idea is not to, you know, no. Well, you'll be fine. And I think after the work that you have done with your unknown, and you know what you continue doing your research and stuff, you'll be fine. But you need to read, you need to study, you need to, you know. And I apologize if sometimes I may sound snappy, but you come at this point in the semester and you tell me, where do I put this? Or what do I put in here? And I'm like, seriously, at this point? And so, because you're supposed to pay attention from the beginning of the semester.